Good morning. Ladies and gentlemen, this witness has been previously sworn to testify truthfully. And if you get to the point, Doctor, where you need to approach the board, please feel free to get up and walk around.
Very good. Thank you, sir.
Mr. Goebel.
Thank you. Dr. Lewine, would you introduce yourself to the jury, please?
Good morning. I'm Dr. Jeffrey David Lewine. I am a neuroscientist by training, so I'm an expert on the brain.
Okay, can you tell me a little bit more about what neuroscience is?
Yeah, so I do have some slides here, although it's not working. Just ignore these for the moment. Just listen to me. They're mostly for me. So neuroscience is the study of the brain, the spinal cord, and the nerves in health and disease. And it includes everything from the basic biology of individual brain cells to how brain cells are interconnected to form networks that support things like memory, attention, the emotional regulation of behavior, to clinical aspects of neuroscience, so how when the brain is damaged, that manifests as disease or injury. And then finally, the techniques that we use to look at the brain. So things like magnetic resonance imaging, MRI, PET imaging, something known as positron emission tomography, and also electroencephalography, EEG. These are all methods that we can use to give us insights into the structure and function of the brain.
Let's talk a little bit about your qualifications to do that type of work and to give those opinions. What's your education background?
Yeah, so I actually have bachelor's, master's, and doctoral degrees in neuroscience from the University of Rochester in upstate New York. I was then a postdoctoral fellow in neuroscience and biophysics at Los Alamos National Laboratory. They got lots of people working on bombs and things, but they actually have a very large biology group, and neuroscience was part of that.
And are there particular areas of neuroscience that you specialize in?
Yeah, so I am generally recognized for my work in different types of imaging technologies. So again, MRI, PET, EEG. I do work on a number of conditions, most notably autism spectrum disorders, traumatic brain injury, epilepsy, and post-traumatic stress disorder.
And where do you currently work?
So I wear lots of different hats depending on what day you find me. So I am the Chief Executive and Chief Scientific Officer of the Center for Advanced Diagnostics, Evaluation, and Therapeutics, located in Albuquerque, New Mexico. Cadet is involved in the development and evaluation of new diagnostic tools and new therapies for autism, PTSD, and traumatic brain injury. I'm also the CEO of Neuroscience Expert Consulting Services of America. That is the small business that is involved in legal work, in this case here. I am an affiliate professor of translational neuroscience at the Mind Research Network, also in Albuquerque. MRN was established by a $60 million grant from the United States Congress to look at brain structure and function in health and disease, especially psychiatric disorders. I have faculty appointments in neurology and psychology at the University of New Mexico, and I'm also the director of research for a small pharmaceutical company, Beyond Barrier Therapeutics, where we're developing and testing new therapies for traumatic brain injury and something known as organophosphate poisoning.
Are you a medical doctor?
So I am not a medical doctor, I'm a PhD.
So you're not here to testify to a diagnosis for Mr. Horner, any particular condition like a doctor would do?
That's correct. So I don't do diagnosis. I'm involved in the science of treatment, but I don't do direct treatment. I don't prescribe medication. I don't do diagnoses for individuals.
Okay, so for this case, I'm here to use your expertise as a brain scientist, for lack of a better word, to talk about Tanner's brain.
Yeah, I was explicitly asked to use the technologies that are available in neuroscience to get an understanding as best as possible of Tanner's brain structure, brain function, and to, based on putting that within the historical context of the information that we have on him, to try and understand what might have caused these abnormalities in brain structure and function.
And is there any kind of independent evidence that you're qualified to integrate the types of information you'll be talking about here today?
Yeah, so I do this type of integration of methods on a daily basis. I have had faculty appointments at numerous universities throughout the country. I've had appointments in the Department of Radiology, appointments in neurology, psychiatry, psychology, even electrical engineering and computer science. So I've got a very broad background in neuroscience and integrating various different types of information.
And do you present your research at scientific meetings?
Yeah, so I have done invited presentations at more than 100 different national and international scientific meetings.
What about publications? Have you been published?
Yeah, so I've got over 75 peer-reviewed publications. I'm the co-author of a textbook on functional brain imaging.
And how is your work funded?
So my work is predominantly funded through research grants. So I've had funding from the Department of Defense, National Science Foundation, National Institutes of Health. I currently have a $2 million contract with Special Operations Command of the Department of Defense for developing and evaluating a new therapy for traumatic brain injury. And just earlier this week, I submitted an application to the Advanced Research and Project Agency for Health for $40 million to examine post-traumatic stress disorder.
Do you do mostly legal work?
So legal work is about 40 to 50% of my effort. Mostly I'm involved in doing these grant types of things. So again, evaluating new therapies, developing new therapies. I do some legal work. It's split between civil work and criminal work like this. About 60% is civil work, 40% of that is the criminal work.
So, have you ever testified as a neuroscience expert in court before?
Yes, so I've testified to my neuroscientific opinions.
You know, I don't want things being displayed here that are not in evidence.
Right.
Okay?
Yes.
Okay, again, this is stuff that's not admitted into evidence. I mean, there's stuff on the board here that is not admitted into evidence.
I think all of that would be on his CV, so we can go ahead and move to his that part.
Will you have any objection?
Well, I mean, it was shown to me, and I wasn't really understanding exactly where they were going with this. None of this is in evidence, and I guess it depends on what they're offering it for.
Well, it appears to be a PowerPoint presentation.
Yes.
So, do you have, you've seen it?
I have seen it.
Do you have any objection?
Well, my objections would be to anything in it that's obviously hearsay, and if it goes to his opinions that are expert in nature outside of his expertise, I would also object to it on that basis. But I don't.
All right, y'all retired to your room. Everyone may be seated. I didn't mean to cut you off, but I mean, he's flicking the deals, and I'm like, wait, what's going on? So.
Well, with the other experts, I'd introduce the CV first, but nothing on here wouldn't be contained on the CV so far.
Well, but I mean, he's testifying, and like this, I've been recently awarded $1.5 million from the Special Operations Command. He's not testifying. I mean, we got stuff up here on the jury that the jury's reading that he's not testifying to, and I want to clear that up. So with this PowerPoint presentation, is there anything in here that the state is going to object to? I mean, I'm going to let him testify to it, but I don't want it being shown to the jury as an exhibit.
Okay. That's the last screen with anything about his career.
Okay.
The rest of it is the materials that we provided yesterday and today.
Okay.
His finding. It's based on the radiology report, the Dr. Bono, and the PET scan, and the EEG scan, and then the quantitative analysis that Lewine did himself.
Okay.
Well, some of the stuff, I mean, we didn't go over all this stuff in the 705, if I remember correctly, but there's stuff in here about he's making assessments as a defendant as to what his toxic exposure is, what it's from, his neurodevelopmental disorders, which he can't diagnose, that comes from somebody else.
Well, but I'm assuming he relied upon the other reports that have been testified to. Is that correct?
Yes, sir.
Okay.
That's correct, but he's not able to make any form of diagnosis and make any conclusions that Mr. Horner is suffering from any of these. He doesn't have that ability.
Well, that's already been testified to, so he can rely upon it.
Ask.
Yeah, I mean, if it's going to be, are they offering it just to be demonstrative, to just show certain things? Or are we trying to admit it actually into evidence? What are we?
This is just demonstrative evidence. We have his 3 reports we're going to move to enter, and we have his CV that's already been entered into the record, and we're going to move to enter it for all purposes.
Okay.
And
Do
then
you have any objection to that?
I don't think so.
Okay.
I had no intention of putting in every, well, I mean, the images he's going to use. I think the report itself probably contains.
As long as we're going to introduce it so the jury can have it, if they deliberate, I'm good
I'll
with
do the
it.
next thing.
Okay. All right.
Is that okay?
Oh yeah, absolutely.
All right. Sorry.
Demonstrative, I mean, I'm going to say demonstrative only on this as far as the power.
Yeah, this is demonstrative only. I don't intend for this to go back to the jury. That's why I supplied it to him.
But we will mark it. We'll introduce it for purposes of the record.
We can absolutely. Sure.
Okay, I'm good with that.
I just want to make sure I understand. So, information about toxic exposures that's in reports that I have read, am I allowed to?
I would
it's-
next ask you to identify these, which will be your 3 reports and your CV.
And that came from Dr.
This is the one.
Speck, correct?
Yes.
Okay. You can rely upon Speck because he has testified in front of the jury.
So Dr. Davies has not testified yet. He did a formal report.
Yes.
Am I allowed to talk about his
Here's one that
observations?
We did a 705 on already.
You are allowed to testify that you received a report from Dr. Davies, but don't go into the report.
Okay.
Okay?
Would that include anything that he's going to testify as potential conditions and results with regard to the defendant that you took into consideration and make it, I mean?
You may
it's all-
testify that you received a report from Dr. Davies that you relied upon, but do not go into anything involving the contents of Dr. Davies' report.
No, but from his perspective, he had, from his neuroscience perspective, what he did, he will be able to say whether his results are consistent with FESD, ASD, those things, but not talk about what Davies is saying.
I'll let him do that.
Does that make sense?
And am I allowed to indicate that Dr. Davies' report indicated that there was likely alcohol
No,
fetal
you're going into the report.
Okay.
Okay?
And some of that's on this.
Well, that's just for demonstrative purposes, so it'll all tie up, so let's go.
Does that make sense?
All right, let's go.
I'm just going to start here, sir.
That's fine.
Thank you, sir.
Yes, sir.
That's what I get for doing something out of order.
I mean, wait, whoa, time out.
Well, Dr. Lewine did offer this all that to say, or, you know, it's like, I just got it.
We're good.
Okay.
All right, everyone may be seated. We're ready to proceed. Mr. Cole.
Dr. Lewine, is Defendant's Exhibit 50 a true and accurate copy of the receiving?
Yes, sir.
Your Honor, that was previously admitted to the record. I move now to admit for all purposes.
Any objection?
No objection to the
State's Exhibit 50 is admitted for all purposes.
Dr. Lewine, is Defendant's Exhibit 71 a true and correct copy of your MRI report?
It's the brain integrity check MRI report, correct?
Okay. Your Honor, I move to admit that at this time.
I'm sorry.
I would move to admit that report at this time.
71, any objections?
No objection.
All right, Defense Exhibit 71 is admitted.
Dr. Lewine, is Defendant's Exhibit 72 a true and accurate copy of the EEG, MEG physiology report?
Yes, in this case, it's just EEG, so the BIC EEG report.
Okay. Your Honor, I move to admit Defendant's Exhibit 72 at this time.
Any objection?
No objections, Your Honor.
Defendant's Exhibit 72 is admitted.
Dr. Lewine, is Defendant's Exhibit 73 a true and accurate copy of the PET report?
It's my PET report, correct?
Yes, Your Honor, I move to admit Defendant's Exhibit 73.
Any objections?
None, Your
Defense Exhibit 73 is admitted.
Thank you, Your Honor. Your Honor, we're going to, one more question. Dr. Lewine, did you review any other materials or records in preparing your report or preparing your testimony?
Yes, so I reviewed extensive records and also a number of expert reports.
I believe I was asking you if you had testified in court before, and you answered that. Have you ever testified in criminal cases in Texas?
Yes, on numerous occasions.
And I believe we have a PowerPoint presentation that we're just going to be using to help explain your testimony to the jury.
Yes, I'm going to go down here and go into a little bit of professor mode here for a little bit. The pointer doesn't work very well, so it's a little bit easier if I'm down here directly. Can you all hear me okay? Perfect, thank you.
All right, so can you start by giving us a little tutorial on neuroscience and what the relationship is between the brain structure, the functioning, and behavior?
Yeah, and so in order to understand what we're seeing in different imaging techniques, I need to give you kind of a little quick tutorial on the brain, how the brain works. And so the brain really is the basis of all complex behavior. It controls how we interact with the world. It controls how we interact with each other. And when we think about the brain, so the brain is composed of different types of cells. The most important one are the neurons. So the neurons are the computational units of the brain. And they basically have three parts to them. They have the cell body, which is the command center. They have the dendrites, which is how signals get into the neuron. Then they have the axon, which is how signals get out to the neuron, go over to the next cell. And so the cells are basically interconnected with a little space here known as the synapse. And your brain has about 80 billion brain cells in it, 80 billion neurons, and about 100 trillion synapses in it. So it's an extraordinarily complex system. And again, it's got the computational units, the cell bodies, and they reside in what's known as the gray matter. And then they have the axons, which interconnect to different cells. That's what's known as the white matter. And so you're going to hear about me talk about gray matter and white matter as we go on. And sir, I apologize, that is my glucose sensor alarm going off, and I thought I turned it off, but I didn't. It's going to beep. Can I go in?
No, you're fine.
Just let it beep?
Yeah.
Okay.
Or can you turn it off?
I can't turn it off.
Why don't you go turn it off?
Okay, my apologies. I thought I had.
Sorry, Judge, it's been one of those mornings.
I agree.
Thank you, sir. My apologies.
Yes, sir. Mr. Coble, you may continue.
Dr. Lewine, can you pick up where you left off with?
So again, there's the computational units, there's the interconnections between them. And so if you damage the gray matter, the computational units, or you damage the white matter interconnections, that's going to disrupt function. And so, for example, the brain actually has some degree of specialization. There are certain parts of the brain that are mostly responsible for visual information processing. Other parts of the brain that are involved in auditory processing. And then there are networks in the brain. So the part of your brain that understands language is in the left temporal lobe. The part of your brain that actually lets you talk and communicate is in the left frontal lobe. And so those are where the computational units are, and then there's axons, white matter pathways, that interconnect those parts of the brain just like the telephone wires. So if you damage either the left temporal lobe, the left frontal lobe, or the interconnections, you get a disruption in language function.
When you're talking about language networks, are there other networks to the brain?
So there are many, many other networks. There are networks for controlling memory. There are networks for doing things like recognizing faces. There are networks for controlling emotion and regulating behavior. And there are also networks in the brain that support what we call social cognition. So this is just a caricature with the areas in yellow showing those parts of the brain that are involved in social cognition. Social cognition is your ability to interact with other people, to recognize their face, to recognize their emotions, to communicate effectively with them, to have empathy for someone. Something known as theory of mind, which is your ability to understand what someone else is thinking. All of those things are controlled by the brain, controlled by specific parts of the brain, interacting with each other.
And is everyone's brain the same?
So, no, everybody's brain, there's a general organization, but the fine details are different. So my brain has basically the same general area in the back of the head that's involved in visual information processing as your area. But my brain is wired a little bit differently than your brain.
Okay, so does that mean that people perceive and interact with the world in different ways because of the way that their brains are different?
Yeah, so there are a couple of things that are important. So one thing to keep in mind is that the brain develops. So the brain you were born with is not the brain that you end with. And there are many, many factors that influence your brain development. Your genetics is important. The environment you grow up in. So if you grow up in a nice, kind, loving environment, your brain actually wires differently from somebody who grows up in what we call a toxic stress environment, where there's not enough food, where there's violence, toxic exposures, alcohol, drug abuse, all of those things, psychiatric disease, and things like head trauma. And so as a result, everybody's brain is different, and this changes how you perceive the world. So I don't know if any of you recognize this, and I don't know how well it's going to show here. This is the infamous yellow and white dress that was on the internet. I look at that dress, and it is yellow and white to me. Both of my daughters, when they look at that dress, it's blue and black. And I don't know if anybody here is seeing blue and black, but about a third of people see this as blue and black. And my daughter's like, Dad, you're crazy. That is white. That is blue and black. And I'm like, no, it's not. It's white and gold. It has to do with the way that my brain wired itself to perceive color and the location of light sources. Much to my chagrin, the actual fibers in the dress are blue and black. I'm misperceiving it. But the point is that how you perceive the world, how you interact with the world, is a function of how your brain is developed. And if neurotypical people can have such widely different perceptions, imagine what happens when you put brain injury and dysfunction on top of that. So how you perceive the world, how you interact with the world, is a function of how your brain is wired and functioning.
Okay, so what factors influence how your brain develops and how it gets wired?
So again, there are all of these different factors that influence things. And so anytime we start to look at brain damage, we start to think about brain dysfunction, and we want to try and understand what the potential causes are, we need to look at all of these different factors.
Now let's talk about Tanner specifically. Can you tell me about some of the things that you've reviewed in his history and records that have at least the potential to disrupt the brain structure and function from your point of view?
So one of the things that I do is I go back and look at medical records, school records, jail records, trying to identify all the things that we know of that potentially cause disruption in brain function. And there are a lot of those, and I try to identify the ones that may be relevant in a particular case, in this case for Tanner. And so, based on these reports that I reviewed, there are certain things that are in the literature there. So he potentially had some toxic exposures. There are some records which suggest that there may have been some fetal alcohol exposure. There are records which indicate that there was probable lead exposure. There are records that indicate that he had this toxic stress, that he grew up in an environment that wasn't the ideal environment for raising a child. And you've heard a little bit about that, you're going to be hearing more about that. He has what we believe are some neurodevelopmental disorders. So you've heard a lot about autism spectrum disorders. And so autism spectrum disorders really are a spectrum. So I do a lot of work with kids with autism. Some of these kids are completely nonverbal, don't make any eye contact.
Judge, I'm going to object to this. This is an extending narrative without any form of question, and it's non-responsive as well to the particular
All
question.
right, sustain. Let's keep it question and answer as much as we can.
Yes, sir. So as part of your work in identifying neurodevelopmental disorders from a neuroscience perspective, have you worked with patients who've had autism spectrum disorder or ADHD?
Yes, so I extensively work with individuals with both autism spectrum disorders and ADHD. And in the records, there's indication that those are present for Mr. Horner.
Hang on.
And
Question.
is head trauma another area that you would look at for Mr. Horner based on his records?
So there's indications that there may have been some head traumas, not well documented in medical records. But again, I consider head trauma as a possibility for something that might disrupt brain structure and function.
And as a neuroscientist, how do you go about evaluating brain structure and function?
So there are a number of techniques that we use. One of the primary ones is something known as magnetic resonance imaging, MRI. So MRI is a technique for looking predominantly at brain structure. There's a big donut-shaped magnet. So you slide somebody into the magnet, and then you actually pulse at them little radio frequency signals. And the system listens for the radio frequency signals that come back. And those signals that come back are influenced by the tissues in the brain.
Now,
So-
those images at the bottom, type of images you would get from an MRI.
Yeah, so these are not Tanner's images, these are just representative images. But so depending on how you pulse the sequences, you can get different types of images. These are what are known as T1 images. They're very good for showing the anatomy. There's something known as DTI, diffusion tensor imaging, which tells us about the white matter interconnections. And one thing that I want to make sure everybody understands, so this isn't like taking a photograph. This is measuring magnetic signals that are coming back from the brain and reconstructing these images. So it's a mathematical, computational thing. It's not like snapping a picture. There are standard ways on how these signals get reconstructed and represented in terms of what the images look like.
And you said there's standard ways, so is this a technology that's widely used in clinical and research and scientific applications?
Yes, so almost every hospital in the United States has an MRI scanner. They all produce basically the same types of images. Not all scanners have the capabilities for the diffusion tensor imaging, but they can all certainly do these types of T1 images.
And this technology has been admitted to courts across the U.S.
Yes, sir.
Did you do an MRI on Tanner?
Yes, so the MRI was actually done at the MRI center. I personally, about a year ago, set up the protocol on the scanner to collect these data. This is just an example of one of the images that comes from Tanner's brain. This is what's known as a sagittal slice, so it's in this direction here. We're a little bit off the midline. The MRI data that were collected, there were two types in particular of relevance in this case. The T1 images, which were used for something known as volumetrics, which I'll explain, DTI, diffusion tensor imaging. These images were provided to a neuroradiologist, and the visual inspection of these images by the neuroradiologist is that they're within normal limits. So the neuroradiologist, just looking at the images, says there are no gross abnormalities here, there's no tumors, there's not a hole in the head, there's not multiple sclerosis.
Okay, let's back up for a second. So you set up protocols, kind of telling what you need the MRI to do, correct?
Correct.
And then a medical doctor, I believe in this case, maybe Dr. Yon, signs an order for the MRI techs to do it. And then it is actually the MRI techs that run the machine. You're not running the machine off.
Correct.
And then they give you the data from it.
Correct.
And you're saying that a normal radiologist reads it and says, when you say there's no gross abnormality.
Judge, I'll object as to leading.
Let me hear the question.
I'm sorry, I thought he already said that the radiologist said there was no gross abnormality. What did the radiologist find?
The radiologist did not indicate any gross structural abnormalities in Tanner's brain.
And we're using the term gross like in the term of a large scale. What do we mean there?
What you can see on visual inspection.
And that's where you said visually you can look at it. He saw no tumors, no holes in the brain, those sorts of things.
That is correct.
Okay. So let's go. You talked about a couple other ways that you, as a neuroscience, analyze it. You mentioned volumetrics. Tell me what volumetrics are.
So one of the challenges with MRI is in children with autism spectrum disorders, even very severe autism, MRI shows things in less than 10%. In mild traumatic brain injury, MRI visually shows less than 10%. What scientists and clinicians have jointly developed over the last 2 decades are what are known as quantitative methods. One of those is a quantitative method known as volumetrics, where we actually measure the size of different brain areas. This is done by a computer. And then the values for the client are compared with average values from a normative database. So just like when you have neuropsych testing or you have your blood work done and they come back and say, hey, your white cell count is done, that's comparing your white cell count with the expected average for people who don't have any conditions. So
Hold it 1 second and explain. You talked about using a normative database. Now, first of all, you said volume, and for a layperson, you're really talking about the size of each part of these brains.
that is correct.
And then you said a normative database. Explain how you create that, what that is.
So again, the normative database is created by MRI images from people without any history of head trauma, neurological disorder, psychiatric disorder, learning disabilities. And so for the work that I do, we're able to compare Tanner's data with a normative database that has about 10,000 people in it. Now from that, we extract only the male subjects who are within 5 years of age, but it's a very large database. And so we can look at the results and say, hey, this is within normal limits, or this value is too big, or this value is too low. And we can do statistical evaluations on it. And all of this is done by the computer. So the computer does the identification of the different brain regions. So that's what these colors are. This is just showing the computer calculating these different brain regions. The computer system doesn't know if it's my brain, your brain, Tanner's brain. The only information the computer has, in this case, is at the time we did the scan, I think Tanner was 33 years old, male subject. That's the only information that it has. So it's a very objective, mathematical, statistical way to determine whether or not there are abnormalities.
And it's making a comparison between his brain, his values, and neurotypical people within 5 years of age. Is that above or below?
Yes.
And that are male.
Correct.
Okay.
And so again, so it's a statistical comparison. So this is just showing a subset here. So the open circles are the values for the database, the neurotypical subjects. And so if Tanner's value is here in green, right smack in the middle, that's within normal limits. If his value is very large compared to the database, that's outside of limits. If it's very small, and we describe how abnormal things are using a statistical measure, something known as a z-score, which basically tells you how abnormal things are.
Okay, and what did Tanner's MRI show?
So Tanner's volumetrics show that things are mostly within normal. There's one particular area in the brain, the left orbital frontal area, that is abnormally low. His value is about 20% lower than what you see in neurotypical subjects. That translates into about 50 million fewer neurons. And again, this here, this underlying brain, this is a caricature based on the calculations. I'm the one who put these colors on and chose the color. So
Okay.
the way you get those colors is the science, and that is standardized. That's how we do it for everybody.
Now, so you're saying that it indicated that he likely had a volume loss, and you were using this to show which part of the brain that would be in. And what does that part of the brain normally do?
And so the left orbital frontal cortex is one of these key brain regions involved in the emotional regulation of behavior and social cognition. It is often disrupted in autism spectrum disorders. Again, that's based on the scientific literature. It can be disrupted in traumatic brain injury. It can also be disrupted in depression.
Okay, and once again, is volumetrics something that just you do, or is that a technique that's widely accepted in clinical and forensic and scientific fields?
Yeah, so volumetrics is an extremely well-accepted technique. It is supported by over 5,000 publications. It is routinely used in hospitals throughout the United States. In the clinical world, it is used mostly in the evaluation of epilepsy and Alzheimer's disease, but it's seeing increasing usage in traumatic brain injury. And certainly in the forensic world, we see it being used very commonly with respect to psychiatric conditions, things like depression, schizophrenia, and autism spectrum disorder.
Okay, let me move on to I think you said you use another technique called diffusion tensor imaging. What is that?
So diffusion tensor imaging, it's still an MRI technique. It's just a different way of pulsing those radio frequency fields to emphasize different characteristics. In particular, the characteristics it emphasizes look at how water is diffusing in the extracellular space. And so remember we talked about axons. So axons travel in bundles. Because they travel in bundles, water within the brain moves along the axons as opposed to across them. And so we can use a measure known as fractional anisotropy, FA, to tell us about the integrity of the axons. And so just like we did with the volumetrics, the computer is going to calculate the FA values, in this case for 33 different fiber tracts, and compare the values for Tanner versus a neurotypical database.
Okay, and what did the DTI for Tanner show?
So the DTI for Tanner shows that most of his fiber tracts look to be intact.
Hold on for 1 second here and tell me about the images. These are the computer images, or you've added color to help us understand?
So these are the images. So the underlying brain here is just a generic one. The color codes here are showing the tractography. So this is the computer's reconstruction of the fiber tracts in Tanner's brain. The color assignment, again, is very, very standard, not something I do, comes out of the computer algorithm. The different colors are telling you about the direction that the fibers are moving in, whether they're going front to back, up, down, or across. And then again, the computer extracts these FA values, compares it to the normative database, tells us whether they're normal, whether they're high, whether they're low. These blue boxes indicate that those fiber tracts had FA values that were abnormally low, in the lowest 5th percentile.
Okay, explain, let's start with what track we're talking about and what they control those values you found.
Yeah. So in particular, Mr. Horner has low FA values for what are known as the right and left fornix. This is a fiber tract that's very important in short-term memory. And the low FA values indicate that there's a disruption in the structure of the fornix.
And did you tell us what the fornix, what did you say?
So the fornix is predominantly involved in short-term memory.
Okay. Now, this DTI, is that a technique that only you were using, or is that also common in clinical and research and scientific settings?
Again, this is also a common technique. Diffusion tensor imaging is used, it's supported by several thousand publications. Tractography is routinely used by hospitals in planning surgery for people with brain tumors and epilepsy. These types of quantitative analyses aren't too often used in the hospital because they don't change medical care, but they are routinely used in forensic and research settings to understand whether or not there is evidence of white matter dysfunction, white matter disruption in conditions like autism, like traumatic brain injury, like substance abuse.
Now, I think you've explained that the volumetrics and the DTI are looking at brain structure. Did you also look at brain functioning, and how would you do that?
Yeah, so looking at structure is one thing, but it's only part of the story. So if you take a group of people with autism spectrum disorders, often, certainly clinically, the MRIs look normal. Even the volumetrics are normal in many of those individuals. You need to actually look at the function of the brain to understand more of what's going on. One of those technologies is something known as electroencephalography. So your brain cells work using electrical currents. So when your brain cells are active, they generate electrical potentials up on the scalp surface, and we can measure those using a special cap. I actually came to Fort Worth. I personally did the EEG exam on Tanner. What comes out from the EEG is a bunch of squiggly lines. These lines are showing how the electrical potential is changing in time at each location. We record from 19 different places over the head.
And are you doing an interpretation on that, or is this a computer?
So this is a computer analysis again. So one of the things that can be done with these signals, so if you have a very complicated squiggly line, you can describe the complicated line by a combination of simpler signals. We basically look at how fast the squiggles are and how big the squiggles are. So the computer can take the actual data and calculate how much what we call power there is in each frequency. Power relates to how big the signals are. And so we get values for Tanner for power at different frequencies at each electrode. Just like we did with the MRI, we can compare that with a normative database.
Now, can you back up for 1 second there? You put up there these different gamma, beta, alpha, theta. What are we talking about there?
So we generally describe, again, the signals by how fast they are. So delta refers to squiggles that are very slow, so between 1 to 4 per second. Beta refers to much faster signals in the range of 20 to 40 per second. And these different types of signals are associated with different types of activity. So delta activity in particular is associated with very significant brain dysfunction. You should not have a lot of delta activity in somebody who's awake. Beta signals are associated with brain hyperexcitability. We see that in anxiety disorders. We see that in autism spectrum disorders. We see that in post-traumatic stress disorders. So by looking at the different frequencies in the scientific literature, we can understand what is or is not abnormal, what it is related to.
The EEG is also a technique that's commonly used clinically, forensically, scientifically.
Yeah, so again, in the clinic, it's used predominantly to look at epilepsy and sleep disorders. Quantitative metrics are used in ICUs to look at epilepsy and stroke. These types of quantitative analyses are also used clinically in guiding a therapy known as neurofeedback. That's typically done more by psychologists as opposed to medical doctors, but still clinical work using these technologies.
And the Q up there, is that for quantitative?
Yes, sir.
And that's just kind of another way of analyzing an EEG?
Yes, so the EEG is the basic data, QEEG is the quantification of the data.
Let's move on, and I think you were going to tell us about what Tanner showed, is that correct?
Yeah, so I just need to tell you a little bit more about how we represent the data. And again, this comes from the computer. It's the same for everybody. The computer doesn't know whose brain it is. These are what are known as the power map. So these are looking at the different frequencies. So again, delta are very slow frequencies, beta are faster frequencies. If it's this very light green color, it means it's within normal limits. And this is not Tanner's brain, this is just a representation here. This particular case had a stroke in the right hemisphere, and so this individual has lots of abnormally increased theta power. That's what the red color is. Again, the computer's assigning these colors. These are standardized. This is using FDA-cleared software for these analyses. There's another measure we can do, something known as coherence, which basically looks at the interconnections between brain areas. And so we do those types of analyses. And so I'm going to show you Tanner's data in a second here. Let me just show you a typical control subject. So even a control subject, because you're making lots and lots of measurements, may have a couple of scattered abnormalities. Okay? That's common. It's known as the problem of multiple comparisons. You can get some false positive things. We have statistical methods for dealing with that. So here's just an example of a control subject. Here's what we actually find for Tanner. So Tanner has very significant increased theta activity all the way through delta and high beta activity over the left hemisphere of his brain. He's got some minor disruptions in coherence. They're actually not that bad. But his baseline brain rhythms are not neurotypical. They show significant abnormalities. One of the other analyses that we can do is to actually figure out where in the brain things are coming from. And again, this is all done by a computer using FDA-cleared software. This is looking at how abnormal the currents are and where those abnormalities are coming from. This is a generic brain, so the underlying brain is not Tanner, it's just for demonstrative. But the colors and the locations of the abnormalities come from the data from Tanner. And he shows in particular abnormal theta activity and abnormal high beta activity over the left hemisphere temporal lobe, parietal lobe, a little bit of the frontal lobe.
And what can we learn from that?
So what these data are indicating are, so that the theta is indicating that there's some general brain dysfunction. Increased theta activity is often associated with traumatic brain injury, but you can also see it in other psychiatric conditions. High beta activity is very consistent with an autism spectrum disorder. Now, you cannot, and I can't look at the EEG in isolation and say, aha, this is autism, okay? These are not diagnostic techniques in that sense. They are supplemental information. These are patterns that are consistent with the presence of an autism spectrum disorder that has been diagnosed by people other than myself.
So you're looking at the brain. We know the brain controls behavior, can give you information on other psychiatric or developmental disorders, but you're not diagnosing solely based on this.
That is correct.
Okay, and what about, are we, yeah, there we go. We talked about this is QEEG, it's routinely used in clinical, forensic, and research settings.
That is correct.
Okay. And what about a PET scan? Was the PET scan done in this one?
Yeah, so the last imaging technique.
First of all, what does PET stand for?
So PET stands for positron emission tomography. So when your brain cells are active, they require glucose. Glucose is the energy the brain cells use. And so PET is a way of looking at metabolism and blood flow within the brain. You actually inject the radioactive glucose, and then the PET scanner images how that glucose is distributed. It does it in conjunction with a CT scan, which is kind of a very fancy X-ray to give you some structural information. So you get the CT structural information, you get the PET information. And I'm just going to show you some things, and I want to be careful to explain this if I may. So the images that get sent to me from the scanner are generally black and white and gray images. They don't have any color on them. There are standard color palettes that are applied to those data to just help to emphasize some things. So that's what the blue and the purples are, to just tell how much metabolic activity there is. So blue was low metabolic activity, yellow was high metabolic activity. Just as we did for the MRI, just as we did for the EEG, the computer algorithms go ahead and calculate whether the values for Tanner are normal or abnormal relative to the normative database. And then the computer goes ahead and puts these blobs on. So these blobs are not inherent in the images. These represent the computer calculations, but they are identifying areas that are abnormal. In this particular case, Tanner has what's known.
Before we get to that, 1 second. Sorry, I missed just legally. So the PET scan, you said that was another one where there has to be, you did a doctor's order, you set up the protocols, an actual techs who do the PET scan did the PET.
That's correct.
Okay, and then was there a separate reading on that?
So there was not a formal reading. The imaging center tells me if there's anything grossly abnormal on the PET. They did not tell me anything that was grossly abnormal. So the clinical read of the PET scan is within normal limits.
And is that once again talking about the same thing? We're looking for the big issues, the hospital issues.
Yeah, for looking at tumors, for looking at Alzheimer's dementia, you don't see gross things on PET in general in psychiatric.
And then you get the data, the computer does its magic science here, and now that's what we're going to talk about.
And so the data actually come to me. I actually send it to a company that does these analyses. So it's like a blood test. It gets sent off to a lab. The lab sends you back the results. And then I interpret those results from a neuroscientific perspective.
Okay, so now can you go ahead and proceed with your interpretation? Sorry.
So he has a number of areas which are known as showing hypometabolism. So these are areas of abnormally low activity. And again, the computer is putting on the color codes here These are standardized codes. This is how they're always used. I don't choose the colors. This is all part of the software. Tanner shows hypometabolism in several areas of the brain, including the orbital frontal cortex, right and left, including parts of the right temporal lobe. He also shows abnormally high metabolism, overactivity. And overactivity is just as bad as underactivity. He shows that in particular in the back of the brain, in a part of the brain known as the cuneus. And then, so one of the things that I have done is taken that information and just for ease of display, color-coded onto a demonstrative map. So blue is showing areas that are hypometabolic, red is showing areas that are overactive.
Now, you mentioned the cuneus there, you had mentioned that before. What does that area of the brain do?
So the cuneus is actually predominantly involved in visual information processing, but it's also involved in social cognition. In particular, things like empathy, and again, this thing known as theory of mind, understanding what somebody else is thinking.
Now, we previously admitted the 3 reports on these 3 techniques you talked about. How would you put them all together? What does that indicate to you?
So when you put everything together, you basically get this. So the left orbital frontal area is actually abnormal on both the MRI and the PET scan. He's got a number of abnormalities on the PET, so right temporal lobe, bilateral orbital frontal areas, the cuneus is overactive, and he's got this general dysfunction in the left hemisphere gleaned mostly from the EEG. And because each of these techniques is looking at different things, it's not uncommon that there's only partial overlap between what you have. But across these techniques, he has evidence of either structural and or functional disruption in many, many areas of the brain.
Great. And can you, and what do these parts of the brain do, and what does all that mean?
So remember that we talked earlier on about this social cognition network, the parts of the brain that let you interact with people? He has wide overlaps between his areas of abnormalities and the social network. So dysfunction, in this case, PET abnormalities in the right temporal lobe, abnormalities in the cuneus, abnormalities in orbital frontal cortex. So he has very extensive overlap with the social cognition network. He has lots of the nodes that aren't functioning properly. You actually need only one node to be damaged to get dysfunction. And so the overall profile here is an indication of objective brain evidence of dysfunction in the social cognitive networks of the brain. From a functional perspective and a neuroscientist, that is most consistent with his history of an autism spectrum disorder. It's not to say that there aren't contributions from other factors like the ADHD, like the toxic exposures, but this pattern is most consistent with social disruption and ADHD.
And can you summarize your overall opinion and what you think may be responsible for the abnormality?
Yeah, so kind of as I just said, he has disruption in EEG, MRI, PET, which collectively show dysfunction and structural disruption in the networks that support emotional regulation of behavior and social cognition, a little bit in some of the memory system, the right temporal lobe, very important for pragmatic language. This is kind of understanding the nuances of language. So he's not, based on the imaging, he's got disruption in those networks. Again, profile most consistent with autism spectrum disorder. Other things might be contributing to this. It's my understanding that Mr. Horner does have a history of some substance use, mostly marijuana, but also some cocaine and hallucinogens. This particular profile is not a good match for those things. It's not to say he hasn't done those things. It's just to say there's not evidence in this particular set of data that his exposure to those substances have caused major permanent brain damage. It's really the autism spectrum disorder that goes hand in hand with these particular findings.
Thank you, Dr. Lewine. You're on pass the
Okay. Let's break for lunch. It's on its way. Should be here in about 5 minutes. So we will start back up at 1:15. Okay? See you then. All right. Everyone may be seated. We are ready to carry on cross-examination. Mr. Staten.
Thank you, Your Honor. Dr. Lewine, thank you for hanging out with us for a little longer this afternoon on a Friday. I've got a few questions for you here. How many times have you testified on behalf of capital murder defendants in Texas?
In Texas specifically, I want to say 7 or 8 times.
Okay. And how many total?
Probably about 15 total.
And in fact, if I remember correctly, you were in Eastland in January doing the same thing.
Yes, sir.
Is that right?
Yes, sir.
Testifying as to the brain abnormalities as of that event, correct?
That is correct, sir.
And I noticed in this, I think you called it your professor. Is that what you called it?
Professor mode, yes.
All right. How many times do you think you've given that spiel being the professor?
I think I've only done it like that 2 or 3 times. I typically do a PowerPoint though.
Okay. Well, you had both today. You had you and the PowerPoint.
Yes.
Now, in this particular case, who hired you in this case?
I was hired by the defense attorney group.
Okay. And who's that?
So Susan Anderson in particular.
And how many times do you think you've worked for them in the past?
Objection, relevant, John.
Over. Go ahead, sir.
I think 4 times. I'm not absolutely certain on the number.
4 times, including the one in Eastland, or just 4 times, including today?
4 times including today. So I don't believe that Ms. Anderson was involved in the Eastland case.
Her organization was involved in that case.
Objection, Your Honor, approach.
Chair will disregard the last question asked by the prosecutor, okay?
Now, in this particular case, you had some scans ordered to be done on Mr. Horner, is that right?
Yes, sir.
Okay, and those were ordered by who?
So I believe that they were ordered by Dr. Yount. I think the PET may have also had an order from a Dr. Rouse.
Okay.
Rouse, R-O-U-S-E.
And in this particular case, just so we get this out of the way, what's your bill in this case?
So my total bill for everything has been about $40,000. That includes the costs for the actual scans.
Okay. And like I think we talked about in this case, you're not a doctor, psychologist, neuropsychologist. You're very clear about that, correct?
Yes, sir.
And you can't make any type of medical diagnosis or conduct any type of psychological or psychological tests.
That is correct.
And in fact, you can't treat or diagnose patients as part of your profession.
That is correct.
So really, in this particular case, you can only really tell us about what you found, but you cannot connect what you found to any particular diagnosis.
Again, I can talk about it in the general sense, as I did, that there are certain brain imaging findings that are consistent with diagnoses. You can't make the diagnosis from the imaging findings alone.
And by looking at something that you see, I'm just going to make sure I'm clarifying this here. You can't say that any particular defendant, including the defendant in this case, has any particular diagnosis just by looking at the scan.
That is correct. So I can't say that he has abnormal brain structure and function, but I cannot do a diagnosis.
All right. Now, the 3 reports that you did in this case, you said an EEG, I'm trying to do them in the same order, MRI and a PET scan, right?
That is correct.
And with regard to the EEG and the PET scan, the MRI and the PET scan, both the doctors who read those indicated that the defendant's brain in this case was functioning within normal limits.
So the clinical interpretation of those scans is that there are no abnormalities there are no gross abnormalities on the MRI. There are no gross metabolic abnormalities on the PET scan. That is correct.
Right. And in fact, if now you don't have any report from the PET scan, right?
I do not. It is possible that the facility did one, but I do not have that.
You got a report from the one on the MRI, correct?
Yes, sir.
And this was done by Dr. How do you say his last name?
Dwayne Buono.
Buono, B-U-O-N-O?
Yes, sir.
Okay. And he's in Florida, right?
Yes, sir.
And I think you said he's a board-certified radiologist?
I believe he's a board-certified neuroradiologist.
Neuroradiologist.
Yeah, I think so. I know he's a board-certified radiologist. I know he did a fellowship in neuroradiology. I'm not sure about the details of the certification there.
All right. And as a result of that, he says things in his report, just going to make sure I get this right, that things are unremarkable, normal, so on and so forth. Is that right?
Yes, and that's very, very common in psychiatric conditions.
But that's what our medical doctor here says.
Yes.
Okay. And in fact, I think that with regard to the MRI and the PET scan, you indicated that most of the things on there were normal and intact. Is that what you said?
Yes, most of the areas we look at are normal and intact. There were several that were abnormal, particularly on the PET.
And I think that we saw, and I'm not going to try to get into the beta, gamma, theta wave thing with you. You said there were some things that looked like they had fast squiggles and slow squiggles.
So those are the definitions of the beta and the delta waves.
Okay.
Yes.
But I think you also said that at some point that with regard to the test, you get false positives on that test at some point, correct?
So on any test, you can get false positives. There's a statistical technique known as a Benjamin-Hochberg correction for multiple comparisons that I also applied to the data. That isolates the ones that are more likely to be true positive versus false positive findings.
And you indicated that the defendant, I think, and I'm not trying to butcher your terminology here, but he had low FA values and that affected short-term memory.
So he has low FA values in the fornix.
Okay.
The fornix typically supports short-term memory. I can't look at the imaging and say there's fornix abnormalities, therefore he must have memory deficits. Again, these are neurobiological correlates of these things. It would be up to somebody doing cognitive testing to say that he actually has those deficits.
Right. And so with regard to that, that's not going to prevent him from having long-term memory that would keep him from understanding that it's wrong not to kill someone.
So generally, fornix damage does not impact long-term memory. That is correct.
And it's not wrong to kidnap someone.
Again, fornix damage would not have him forget that information.
Now, when I'm looking at all these abnormalities that you're talking about, and you described a whole lot of stuff, okay, and I'm not going to try to talk about all the technology behind it, but how is it with all of this stuff that the defendant is not just sitting in a room drooling somewhere?
Sure. Yeah, so there are levels of abnormalities. These techniques are very, very sensitive. This profile of abnormalities is still on the relatively mild side. So these are the types of abnormalities that we see in individuals with psychiatric dysfunction, with things like autism spectrum disorders. The fact that he has a lot of them, doesn't mean he's a fully demented individual.
Okay. And I guess what I'm asking is that, in this case, I guess what I'm asking, how is it possible that he can function, for example, drive a car, read a map, things of that nature, if he has all of these abnormalities and things that you're talking about?
So again, it matters where the abnormalities are. His abnormalities are predominantly in the social cognition system. So he has orbital frontal cortex compromise, but he doesn't have compromise of dorsolateral prefrontal cortex. He doesn't have compromise in the brain areas that support the types of skills that you just mentioned.
So, but with regard to what you're talking about today, you haven't watched the videos in this case, have you?
No, sir.
You didn't watch any of the evidence as far as what happened in this case?
No, sir.
So you can't comment as to whether or not he has any specific real-world deficits. You can't make those comments.
I can't from the imaging. I can say that people in general with these profiles have deficits in social cognition. I cannot specifically say that Mr. Horner has deficits in social cognition other than from the information that's in the record. So the diagnosis of autism spectrum disorder, the core feature of that is social cognition disruption.
Now let's talk a little bit about, we talk a lot about the human brain, okay? And you'll agree with me that the human brain has an amazing ability to adapt and function despite many factors that come into play.
It can, yes.
Right? And when we talk about, you gave an example, I think, of all the things you compare your test brain to. What was that called? The sample of all the people?
The neurotypical database.
Neurotypical data. And there's not really anything such as a normal brain. Nobody has the same brain, do they?
Not exactly the same. So the neurotypical database takes into account the normal variability.
And it is possible that everyone has potentially some form of abnormality going on, but that doesn't mean they have any deficits.
So again, what you would see is, depending on how many things you measure, you would see that an individual subject might have one or two abnormalities. Again, this is this multiple comparison problem. We use the Benjamini-Hochberg to address that issue. So the number of abnormalities that Mr. Horner has, we would see in less than 0.01% of the population.
So even with any abnormality and even with some deficits, you're not telling us that a person is going to act a certain way?
Again, I can only talk about the general, this is what it's correlated with. From the imaging alone, I cannot say anything specific about Mr. Horner's behavioral pattern.
I'm talking about anyone. You can't say any person will act a certain way.
Not explicitly, that is
You
correct.
can't say whether or not they'll commit crimes.
Correct.
And you can't say whether or not they'll engage in any particular conduct.
Correct.
And for example, I mean, we have individuals out there, let's say, my example I thought of was football players. You'd agree with me that they may have some brain injury at some point. Would you agree with that?
Many of them, yes.
But that doesn't automatically mean that they have deficits, right?
It doesn't explicitly and automatically mean that. It means that they are much more likely to, but for an individual person, I can't say, hey, I've got this finding, therefore this person must have this deficit. You can't do that from the imaging.
Right, and you said you got a grant from our special operations warfighters, correct?
Correct.
And in that particular field, we've got a lot of military service members and veterans that may have some abnormality due to a combat injury. Would you agree with that?
Yes, sir.
And that doesn't necessarily mean that they're going to be somehow unable to continue defending our country.
Again, not necessarily, but on average, they have compromised skills that compromise operational readiness, which is why SOCOM is funding us to look for therapeutics.
Now let's break this down a little bit. We've been talking about the brain. Will you agree with me that the brain and a person's mind are different?
There's lots of theoretical work on that. That's a philosophical debate. I will say that the way that one's mind interacts with the world is through the brain. And so if your brain is damaged, how your mind is going to interact with the world is highly likely to be compromised.
But your mind, you'll agree with me, is where we get the person's reasoning, motive, things of that nature, right?
But it's all filtered through the brain. So I see that dress as yellow and white. I cannot free will myself to think that that dress is black and blue. And so the way that your mind, if you want to call it that, perceives the world, interacts with the world, is through the brain. So if your brain is damaged, the information going into your mind, the information coming out of your mind is going to be compromised.
And so when you're talking about that, use the term free will. Your testing's not going to be able to determine free will in an individual, does it?
No, absolutely not.
And then neuroscience really doesn't have anything to do with person's free will, does it?
Again, there's lots of neuroscience written about free will and mental status.
But your neuroscience, you're not able to distinguish between those who cannot control their impulses and the people who choose not to control their impulses.
I cannot clearly distinguish between that. That is correct.
How many brains do you think you've scanned?
Across conditions, I've probably been involved in the evaluation of about 7,000 brains.
And the stuff you're looking at, what we're talking about today, Mr. Horner's brain, right? This is not the first time you've seen stuff like that before.
It's not. This is one of the rarer occasions where there are so many nodes specifically within the social cognition network, but I've seen other abnormalities, again, more commonly related to things like TBI as opposed to autism spectrum disorder.
And you were in here and you heard the last witness in here, did you not? The nice speech therapy lady?
Yes, sir.
And you heard from her that she said that autism did not cause this crime. You can't disagree with that.
I do not disagree with that.
That's what it is.
Redirect.
Judge, I will have nothing further. Thank you.
May Dr. Lewine be excused finally?
You may.
Thank you, sir. Our next witness will be testifying remotely, so I need a few minutes to set up everything, okay? Okay, everybody, give me a few minutes. I need the state's HDMI cable.